Dear colleagues, it's my great pleasure in these 15 minutes to discuss with you some data about immunotherapy in advanced lung cancer. The question, and that is the one I will address, is a bunch of new data. Is newer data of value or just repetition? So you have understood, I will focus on the 2021 new data and try to put them in the context. This is my disclosure slide. First of all, remember about new data, new strategy, new compounds, that we now have identified some of the parameters which dictate immunotherapy sensitivity. For example, high PD-L1, high TMB, interferon gamma expression, or immunotherapy resistance, for example, Ioio activity, adenosine pathway, VEGF. So we know more and more about potential biomarkers and potential new strategies. That's a glimpse of hope. We also put on this map of the immune cycle. Remember immune cycle with all of the steps to develop an anti-cancer response. We put new compounds on the map. This is the updated one. So we have hopes for new combinations, new data, which will really change the landscape. Is it already the case? Well, you'll see, at least not so much. So first of all, looking at immunotherapy in lung cancer, is still basically focusing on two synapses. One during the priming phase of the immune response, the CTLA-4, one during the effector phase of the immune response, the PD-1, PD-L1, with a series of compounds, two anti-CTLA-4, but a growing list of anti-PD-1 on the top or anti-PD-L1 on the bottom. So the question, first of all, we've seen many data with new anti-PD-1, anti-PD-L1 last year. Does it really change the game? So very importantly, are there biological differences between new and old anti-PD-1 and anti-PD-L1? The answer is yes, of course. There are biological differences. They can be IgG1, they can be IgG4. They have different half-lives, as you can see, and they are characterized by different, I would say, affinity for the Fc gamma receptors, remaining leading to antibody-directed cell cytotoxicity or complement-directed cytotoxicity. So there are biological differences. They will differ in terms of level of PD-1 occupancy, volume of distribution, toxicity profile, and as I said before, serum half-life. However, today, in the practice, in the daily practice, I must state that none of these biological differences has translated into a real outcome difference. So today, this is more a repetition and not really, I would say, an innovative context or concept to be taken over with this new anti-PD-1, anti-PD-L1. So that's where we were basically at the beginning of 2022, with, as you can see, on the left-hand side, non-squamous, on the right-hand side, squamous, and a series of options, IO chemo, IO-IO, IO-IO chemo, and in high PD-L1, IO monotherapy. Of course, all of these schedules has been completed by this new anti-PD-1, anti-PD-L1. You can see that the market is now defining a market for them and across many diseases in various continents. But as I said before, I don't think it will change the science and the outcome, but it might change the accessibility. So in terms, and this is an example of in frontline therapy, how this new cemiplimab, camrelizumab, sintilimab, tislelizumab, sugemalimab, sorry for all these names, have reproduced, repeated, mirrored what was existing before in other trials. So no real advances, but maybe a larger accessibility. So what I'd like to discuss with you today is maybe some things which have changed. So what is important for me in this graph is in yellow, Empower Lung 3, Checkmate 9LA, and Poseidon. And I'd like to discuss these three trials, which I think is not repetition. So first of all, let's speak about this standards of care we have. Usually you use chemotherapy plus checkpoint inhibitors. You remember that this is a standard option in adenocarcinoma based on 3 trials, adding pembro, adding atezobev, or adding atezolizumab, right? And this is the kind of curve you observe as a standard of care with chemo IO in non-small cell lung cancer versus chemo frontline. So this is a backbone of what we do. And this remains valid across all strata of PD-1 expression, negative, intermediate, or high. So that's one of the option standard that we have to keep in mind because this is the comparator. We have the same data in squamous non-small cell lung cancer, but I just would like to say that it's only one trial positive with pembrolizumab again. And I wanted just you to look at how the standard defined differences in survival. But if you look at the difference in survival in negative PD-L1 patients, you will agree with me on the right-hand side that these negative PD-L1 patients benefit, I would say, marginally or at least less from the combination of chemo and IO. I just wanted you to keep it in mind because of the changes we will bring with the next trials. So first of all, the Empower Lung 3 was a good trial because it was addressing the addition of an anti-PD-1, cemiplimab, to chemo in all histologies. So meaning in all incoming non-small cell lung cancer with no oncogene addiction, whatever PD-L1, you could treat them with cemiplimab plus chemo, with all the potential backbones of chemo that we usually use. So it's a very real world clinical trial. And what is good in that trial, it doesn't really change, but it tells us that IO has to be added in all coming non-small cell lung cancer, and that they can reproduce what was partially represented across previous trials in a large trial without selection. And as you can see, a very strong confirmatory trial. What is interesting here is, again, what I said to you before. I want you to look at this waterfall plot for one thing, is the confirmation that negative PD-L1 patients only marginally benefit from IO chemo versus chemo, and we need to do better there, right? The other thing here, which is important to remember, is the addition of IO is not a very strong efficacy in never smokers. So maybe new data because it reproduces, it goes for all lung cancer, and very importantly, we still struggle with this negative PD-L1. This is the data of Empower Lung 3 put in the table, so you can have them, you can look at them, and that's how this new data was presented. But now, what I would oppose to this skeleton is the IO chemo trials. So the first of this one is 9LA. 9LA is IO, EP-Nivo, remember EP low dose, and chemo, short duration chemo, only 2 cycles. So Nivo EP for 2 years maximum, and only 2 cycles of chemo versus chemo. All patients, regardless of PD-L1, could be enrolled without actionable alteration. Primary endpoint OS. What is good, 2 cycles of chemo, overall survival benefit for Nivo EP chemo versus chemo, hazard ratio 0.72, and no early deaths, so no risk of early progression when you give 2 cycles of chemo. So now look at my subgroups when you use IO. So first of all, PD-L1 negative on the bottom of the slide look like to be recovered by the use of CTLA-4. Jim Allison had shown it before. The CTLA-4 induces expression of PD-L1. Importantly, it looks to have an incremental effect as compared to the control arm in patients with CNS mets. So potentially really having a specific activity CTLA-4 when you have brain mets. And as you can see here, again, the never smoker have a difficult story. Keep in mind here that squamous and non-squamous benefit, but if anything, squamous is slightly better than non-squamous, but let's say they both benefit, okay? The activity is therefore, as you understood, conserved in negative PD-L1 tumors, so way better than chemo IO, and it might be a way to move. Why would it be the case? Potentially, somehow you have to keep in mind from melanoma and now 7 papers that when you give CTLA-4, you broaden the repertoire of T cells. So it might be that, first of all, you encompass the recognizable feature of some new neoantigen, maybe in the brain, maybe in PD-1 negative tumors, maybe in general, potentially leading to memory T cells and a longer duration of treatment. And when you look at the brain, look here at the curve. This is overall survival in patients with or without brain mets in 9LA. You can look how much there is an incremental, another layer of activity in patients with baseline brain mets. In that trial, they were treated and asymptomatic. And as you can see here, very impressive to see the disease control rate at the brain level in these patients with the CTLA-4. And I love this intracranial PFS, which leads to a hazard ratio of 0.736 with Nivo-IP chemo versus chemo. So really something in the CTLA-4 here. The second trial, which was a change last year, is Poseidon, where it's the same strategy of platinum-based chemo as a control arm and an arm with durva-tremi chemo. There was a primary arm of durva plus chemo, but as you can see, a very complex statistical strategy, which was mainly basing on the durvalumab plus chemo. But unfortunately, this arm failed. If you look on the right-hand side, the durva plus chemo versus chemo failed, but there was still an option to continue with 1% of the alpha for durva-tremi chemo versus chemo. You can see at the level 2, which was further conducted. So the usual chemo IO failed, but the chemo IO was successful. It was, be careful, a secondary endpoint. So this is durva chemo. As you can see here, the PFS was improved, but not the OS. So we can say it failed because you want nowadays an OS benefit. But when you see at the durva-tremi chemo versus chemo, here PFS is significant and OS is significant. You don't have early deaths, which is a progress as compared to many IO trials. And I want you now to look at my advances, because now you can compare durva chemo to durva-tremi chemo. So if you look at negative PD-L1, TC less than 1. So when you use durva chemo versus chemo, you don't have benefit, like I said before. When you add the tremelimumab, maybe because of the expression of PD-L1, maybe because of the neoantigen recognition, you save this subgroup of negative PD-L1. That's very important. And again, in that trial, you can see the other subgroups as before. But interestingly, in that trial, squamous were doing slightly worse. Let's go into this graph. This is OS in non-squamous and in squamous. And you can see that in squamous, it's difficult to decipher a benefit of the 4 drugs regimen versus chemo. Difficult to understand why. The gemcitabine treated patients were not doing well with durva-tremi, but still to be understood. But basically, an interesting trial to add to our data set. Advances, real advances, have come from CTLA-4. Brain, negative PD-L1, and who knows, based on melanoma, maybe duration of benefits. But the real advances will come from innovative IO agents. And here's a wonderful review telling all of the mechanisms we have in terms of new drug to try to further activate the immune system, starting by the chemokine manipulation, but also all the microenvironment modulation, metabolism inhibitors, immune effector cell modulators. So really something which is now moving in phase 1 forward. This is the usual review in Nature about how the market of drugs is developing. And you can see that based on a skeleton of anti-PD-1, anti-PD-L1, there is a wide range of new compounds which are now used to try to really improve the combinatorial opportunities in non-small cell lung cancer. This is a list of the upcoming phase 3 trial with novel agents in frontline metastatic non-small cell lung cancer, adding the anti-IL-1 beta canakinumab to pembro chemo, adding PARP inhibition, trying to reach antithetic lethalities using olaparib or niraparib, adding anti-angiogenic lenvatinib, or adding anti-TIGIT in high PD-L1 tiragolumab. So these trials will come and will be very interesting to improve our knowledge. And with this, I thank you for your wonderful attention.
Thank you so much, Dr. Solange Peters, for a wonderful lecture. And as usual, we had many of those interesting things to pick up from.
Yeah, go ahead.
So we had some queries, so I'm sure you take it up. So when we want to use these monoclonal antibodies, which test does it matter, or we can exchange anyone? Till recently we were comfortable, we had one drug, various ways of using it, but you did show that we need to be a bit careful about it.
Yeah, so basically we have been doing this effort. We have been pioneers in lung cancer to try to understand how to measure the expression of PD-L1. We've been struggling a lot, probably for 2 years, trying to establish how to do that. Because as you said, every company was coming with their drug and their test. So it was a huge mess. So what we did in the ISLC, the International Association for the Study of Lung Cancer, with the pathology group, we made this blueprint project, which was a way to compare how you can score with one assay as compared to the other one, the one from Roche as compared to the one from AstraZeneca, and so on and so forth. And basically the answers is they are more or less all comparable. So they are all more or less all superimposable. But basically, if you really look at how standards have been reached, you need to make sure that you use a test which is validated. So they are comparable. You can use one for the other one. Particularly now is the 22C3 and the 263, which are used, right? But there might be other ones because remember in the world, many centers use their own test, what we call the laboratory developed test, right? The LDTs. So if you use your test, please compare it to a standard test initially to make sure it's okay. But basically, all in all, PD-L1 is heterogeneous and dynamic. So if you have a good test well compared to 22C3 or 263, then you can move with your LDT, your laboratory developed test, without any problem. So I think we have gone through this struggle of testing by establishing good standards.
The next question from my side will be regarding brain mets. That data is again looks very good. What could lead to that? So a Nivo EP in PD-L1, you explained the reasoning. What could be the hypothesis that in brain mets that difference was very good, and as compared to what we have seen otherwise?
Yeah, it's quite, I agree with you. It's quite amazing, right? Because it's the same in melanoma. Remember that Georgina Long has shown in many trials. The last one was at ASCO this year, 21, the ABC trial, where Nivo EP is way better than Nivo in brain mets of melanoma. So there's really something correct across diseases, right? So what is probably the hypothesis is two things. First of all, maybe by enlarging the diversity, the repertoire of T cells, you might recognize a larger number of cancer cells. So just by making the sensitivity of the immune system higher, you might improve even in niches like the brain, which is something difficult. Remember, the drug doesn't pass the blood-brain barrier. Only the T cells can travel. So maybe you make travel a more diverse population of T cells if you have CTLA-4. But it is a debatable kind of argument. I would rather think that using CTLA-4 might at the end change the microenvironment. And remember that the brain has a very strange microenvironment around the tumor cell. Just think about the antigen presenting cells, which are the Langerhans cells, right? So probably the whole CTLA-4 concept is changing the paradigm of the microenvironment of the tumor, making the brain more sensitive to immunotherapy. Remember, on the other hand, that glioma were never reachable by any IO. So there's something in the brain which makes this environment not favorable to immunotherapy. And this might be the microenvironment. So
you think maybe if they use combination in glioma, they have a chance?
They are trying, right? They are now trying to do more and more combinations. Glioma, of course, remember, is also a lower neoantigen load, but it's really difficult. Remember, in glioma, the difficulty is you need to mount the immune response in a tumor which is located in the brain. When you have a lung cancer, you mount the immune response in the lymph node, in the tumor. So the drug is getting there very well in your lymph nodes, in the lung tumor. So you can mount a T cell response. When you think about the brain, the EP and the Nivo does not really reach the place where the tumor is because it doesn't cross the blood-brain barrier. So it's really a challenge, right?
Absolutely, I agree with you. So next question is, and what you showed consistently, that if a PD-L1 expression is not there, looks like alone this drug doesn't work. Not based on approval. What is your thought process on second line Nivo? Do you think it's the a waste of giving it, in PD-L1 non-expression?
Well, I don't think it's a risk. Well, PD-L1 negative, everything you do is less efficient. You could not say it's absolutely inefficient, because remember, PD-L1 is somehow heterogeneous. So it might be, well, when you have PD-L1 negative tumors, remember the 3 phase 3 trials in second line, even negative PD-L1 look like extracting some benefit, less, but some benefit. Is the reason that you missed some PD-L1 somewhere else in the tumor, or is it because there are other parameters, TMB, Tregs, MD, myeloid-derived suppressor cells? I don't know. But PD-L1 negative have a benefit, but a smaller benefit of the strategy. But I guess you need to try, because even in PD-L1 negative, you will end up with 5 to 10% of your patients telling you 10 years later down the road that you told him he will die or she will die. So, 10% long-term survival is something. But I agree with you, the probability is lower, but you still need to try. What is good, and what Jim Allison has shown before the Nobel Prize, is if you have a chance to give anti-CTLA-4, it will make an interferon gamma signature exist, meaning the PD-L1 expression exists. So that's where the combination might be interesting.
Thank you. And my next question will be, have we given up beyond KRAS for mutation positive patients for immunotherapy, or you think, no, we will be getting something on, as the time goes by, initially we thought it's only EGFR, then we now ALK is also something where immunotherapy is pretty questionable, ROS1, or are they differential, or do we put in the same basket apart from KRAS?
Yeah, it's a very, very good question. I would say KRAS is a good candidate. Remember, if anything, when you have a KRAS mutations, it's usually a smoker patient, so you have a very strong efficacy of IO. So KRAS is a good player. But the other ones, RET, ROS, MET, BRAF, HER2, ALK, and EGFR, are usually found in never smokers. And never smokers are patients who present with a low neoantigen burden. So probably they are all in the same bag because of biology. So biology is a very small number of neoantigen. However, remember, we have some data that if you have a BRAF mutated patient who is a smoker, or if you have a MET exon 14 skipping mutated patient who is a smoker, that suddenly immunotherapy might work. So there's really something about this never smoker story, right? Which puts all of them in the same bag. Have we given up? Well, again and again, it's not that there is zero activity, but you need to divide the activity by 2 or 3. So instead of having 20% response rate, it's probably more between 5 and 10% response rate in these patients. And the gain in terms of OS and PFS becomes pretty marginal if you think about sustainability of our health systems. So I would say we have not given up, but immunotherapy is not an early line strategy because the magnitude of benefit is very small. However, I'd like to rediscuss the EGFR, because of course EGFR is TKI, maybe amivantamab if you are in a country where there is a trial. There are many lines of targeted therapy. But remember that there was this IAMPOWER 150, which was showing that specifically in EGFR mutated, giving chemo, IO, and bevacizumab was better than chemo, but was also better than chemo and bevacizumab, or better than chemo and IO. So really the combination of IO and bevacizumab being interesting in that disease. It was really put into question because it was an exploratory subgroup. It was retrospectively done. So it was questioned by many people. But remember, at the ESMO plenary 2 weeks ago, we had the Orient 31. It's a trial by Dr. Liu, remember, from Asia, who was showing again the same thing, right? Trying to use sintilimab, pemetrexed, and an anti-PD-1. They was able to show that basically, again, IO anti-angiogenic sorry, an anti-angiogenic. IO and it's called IBI305. This is the anti-angiogenic. Sintilimab plus an anti-angiogenic plus chemo is really giving an option in EGFR mutated. So I'm start to really believe, I don't know about you, that in EGFR, IO and anti-angiogenic might be the way to move. So now I'm pretty convinced, and this data from the Orient trial is pretty amazing, right? So maybe there's something to explore, and maybe we'll get some other things for other diseases in the future. What do you think?
Yeah, I think so. I agree with you because the kind of disease we deal with, we all want to stick to whatever little hopes we get, and sometimes that hope becomes a bigger one. I'll take a minute or two of more of yours. One is that we do have now good data with PDL1 and then CTLF4. Do you think the other, which we have heard a lot, that's a LAG3, TIGIT, all other checkpoints? What kind of data we have, or do we think that it is going to take time?
Yeah, it's going to take time. What I mean by that is LAG3 is now in investigation. You remember it was breaking news in melanoma, right? It works very well in melanoma. By the way, it competes with CTLA4 because it's so good that it's a real competition, but it's ongoing in lung, right? In lung, there are trials ongoing with chemo plus IO plus LAG3. Remember, every company has LAG3. So this data will come. We are more at the step of feasibility, but they will come. For TIGIT, it's the same thing. We have trials with chemo, Atizo-TIGIT, or we have trials with Atizo-TIGIT, which are ongoing and should read in the next 2 years. So we are just a little late, but I still hope really that these other, we can call them other checkpoints, right? These other checkpoints might be good ways to move in lung cancer too. So I'm pretty, I would say, optimistic about the potential use in lung cancer. We are just a little late. What was a little failure was the cytokines. We've been moving a lot with, trying to manage the cytokines with pegilodecacin, canakinumab, and so on and so forth. And at the time being, none of this cytokines manipulation did work. So that's maybe one thing which we are weaker than we thought. It's about how cytokines can really change the environment.
One last question. The neoadjuvant data, I remember you discussing about it, and you were very excited, and I totally agree. 3 cycles, good enough, especially we coming from developing country. Are we giving immunotherapy more than required? Never ending, and the patient gets fatigued.
Yeah.
That 3 cycles, the data, event-free survival data is not out, but company has put in press release, looks good, looks positive.
Absolutely. So no, I fully agree with you. We might never have the answer for advanced disease because it's not in the advantage of the pharma industry to show that you can make it short. But and if you agree, probably if we had to do a trial, we should differentiate the patients who respond to the ones who have stable disease, because I'm pretty sure that having a very good response or a complete response defines a group of patients in which you could stop quite early, like in melanoma. Remember, in melanoma, when they reach complete response, they do 1 or 2 more cycles and they stop. So we might also do something like that at some point, but I'm not sure we will have this data at any time point. So in countries where sustainability might not be reached, I would rather say that stopping is not the way to move, but the interval might really be prolonged. Remember the phase 1 from Julie Brammer in the JSU in 2015. She was really showing that after 2 infusions, you have a pharmacodynamic plateau for 100 or 200 days. So instead of stopping, I would rather make the interval longer if you cannot afford the 2 years of IO. So I really think that we are more home brewing here than evidence-based, and we might never have evidence-based.
Then maybe I'll add to one question, and the real last one. Any want to comment about dosing? So good, you commented about the interval part. So people getting ideas about dosing is also a good idea to explore. That
Yeah,
maybe
I think- yeah.
rather than giving the dose what we are giving today, maybe 1/10 of the dose or 1/4 of the dose, or
Yeah, these are all the things we should do. What we could do as academic groups at some point would maybe not be to do big trials with 400 participants, because we will not be able to finance them. But we could try to do good pharmacokinetic trials, right? Trying to understand at each cycle, like pharmacokinetics can be do, pharmacodynamics, what it makes to give half of the dose, or what it makes to give full dose every 8 weeks. So I think as academic groups, what we could move to is carefully try now to do real pharmacology, because pharmacology at the end is the basis of everything, right? If the drug is here, then you can really understand how it acts.
It was a really nice presentation, Dr. Peter. Solange, any other last comment from your side?
No, thanks a lot for having me. It's a great pleasure to meet you.
Same here. Thank you so much. I enjoyed your talk. I'm sure all our
Thanks
audience.
a lot for having me. Bye-bye.
Thank you. Bye.